Good morning, everyone. I'm delighted to welcome you all today. Today's presentation is titled Accuracy Criteria in Analytical Method Validation. I'll be guiding you through the key aspects of ensuring accuracy in analytical methods, which is crucial for reliable results. We'll cover the essential criteria, best practices, and how to validate methods effectively. So let's get started and explore this important topic together. Welcome to our discussion on analytical method validation. As outlined in USB 1225, which covers validation of compendial procedures, there are several key parameters we need to consider when validating an analytical method. These include accuracy, which tells us how close our measurements are to the true value. Precision, or reproducibility, helps ensure our method produces consistent results under the same conditions. Specificity is about the method's ability to measure the analyte of interest accurately without interference from other components. Linearity confirms that our response is proportional to the concentration over a specified range. Range defines the interval between the upper and lower levels of analyte that the method can accurately measure. Detection limit indicates the smallest amount we can detect, while quantitation limit is the smallest amount we can reliably quantify. Robustness tests how well the method performs under small, deliberate variations in conditions. Now, why do we go through all this? Analytical method validation is essential because it ensures our methods are reliable, meaning they consistently produce accurate results. It guarantees reproducibility, so different analysts or labs can get similar outcomes. And most importantly, it confirms the method is suitable for its intended purpose, whether that's for quality control, stability testing, or other applications. All these aspects come together to build confidence in our analytical results, which is fundamental to maintaining quality and compliance in our work. Now, let's talk about how we evaluate accuracy in our analytical methods. Accuracy is assessed across several key areas: assay methods, impurity methods, dissolution methods, and content uniformity. Each of these plays a crucial role in ensuring the reliability of our results. Typically, we determine accuracy through recovery studies, often using what's called a spiking method. This involves adding a known amount of analyte to the sample and then measuring how much we can recover. According to the USB, accuracy is defined as the closeness of agreement between the value we find and the true or accepted reference value. To put it simply, it's all about how close our measured result is to the actual amount we're trying to determine. This concept is fundamental because it directly impacts the validity and trustworthiness of our analytical results, which ultimately affects product quality and safety. So, understanding and properly assessing accuracy ensures we're providing precise and reliable data in our work. Let's take a look at the levels used for the accuracy study in the USB practice. For the assay, we typically evaluate at 3 different levels: 80%, 100%, and 120%. These levels help us assess how accurate the method is across a range of concentrations. It's important to note that we set a minimum of 3 levels, with 3 replicates at each level, which ensures the reliability of our results. Overall, this means a total of 9 determinations are performed for the accuracy assessment. Now, how do we determine the accuracy? We use what's called a recovery study, or the standard addition method. Essentially, we add a known quantity of the standard drug into either a placebo or the sample matrix. Then, we analyze this mixture using our chosen method. The key point here is that we calculate the percentage recovery, which tells us how much of the standard we can recover from the sample. This percentage indicates the method's accuracy. The closer to 100%, the better. By understanding these levels and the recovery process, we can confidently evaluate how precise and accurate our analytical method is, ensuring the quality and reliability of our results in the overall testing process. Today, I want to talk about the assay accuracy study, which is a crucial part of our validation process. Essentially, this study is designed to evaluate how accurate our testing method is. We want to ensure that our assay produces reliable and precise results, which is fundamental for maintaining confidence in our data. During the study, we compare our assay results against known standards or reference methods. This helps us identify any discrepancies and assess the overall performance. The key points here are accuracy, reproducibility, and consistency across different samples and conditions. By thoroughly analyzing these factors, we can confirm that our assay is suitable for its intended purpose. This validation step connects directly to our broader goal of delivering high-quality, dependable testing solutions. In the end, the assay accuracy study is about building trust in our methods, trust in our results, and ultimately, trust in the insights we provide. Let's talk about acceptance criteria, which are essential for ensuring quality in our USP guideline practice. Typically, for assay results, we look for acceptance ranges between 98% and 102%. This means that when we're measuring the potency of a compound, we want it to be very close to 100%, allowing a small margin of error. For impurities, the acceptance criteria are a bit broader, usually ranging from 80% to 120%. However, it's important to note that the exact limits can vary depending on our internal standard operating procedures or specific regulatory guidance. Always double-check those to ensure compliance. Now, why do we set these specific ranges? They help us determine whether a product meets quality standards and is safe for use. Finally, when we talk about the mean percentage recovery calculation, this is the method we use to assess how accurately we're measuring our analytes. It involves calculating the average recovery percentage across multiple tests to ensure consistency and reliability in our results. This overall approach helps us maintain high-quality standards throughout our processes. Let's take a closer look at the accuracy for the impurity method. This approach is crucial because it helps us understand how well our impurity measurements reflect the true composition of the sample. Essentially, we're checking the precision of our impurity detection techniques. The key point here is that high accuracy means our method consistently provides measurements close to the actual impurity levels. This is important because, throughout our presentation, we've emphasized the need for reliable and repeatable results in chemical analysis. When the impurity method is accurate, it boosts our confidence in the data, enabling better decision-making and quality control. So, in summary, assessing accuracy isn't just about numbers. It's about ensuring that our impurity assessments are trustworthy and that they truly represent what's in our samples. A method can be either precise but not accurate, or accurate but not precise, and understanding this distinction is crucial. When I say a measurement is precise but not accurate, it means that the results are consistently close to each other but not close to the true value. Think of repeatedly hitting the same wrong spot on a dartboard. On the other hand, accurate but not precise means the measurements are close to the true value overall, but they vary a lot from one measurement to another. Imagine darts landing near the bullseye, but scattered all over the board. Recognizing the difference between accuracy and precision helps us evaluate the reliability of our methods and understand how to improve them. This distinction is key when we aim for both consistency and correctness in our results, which ultimately supports the integrity of our data and conclusions. Let's talk about the importance of accuracy, especially in our field. Ensuring correct dosage in pharmaceutical products is absolutely critical. If the dosage is off, it can lead to ineffective treatment or even harm to the patient. Accuracy also helps us prevent under- or overestimation, which can have serious health implications. Moreover, precision in measurements is required for regulatory approval. Regulatory bodies scrutinize these details very closely. Without it, a product simply can't get approved. It all ties back to patient safety. Accurate measurements mean safer outcomes for those relying on our medicines. Finally, accuracy is a cornerstone of quality control. It helps us maintain high standards consistently, ensuring that every batch meets the necessary quality criteria. So, in our overall goal of producing safe, effective, and reliable pharmaceutical products, accuracy truly is the foundation. Let's wrap up with the key takeaway on accuracy. As highlighted in USB 1225, accuracy is a critical validation parameter that ensures our analytical methods are dependable. It's typically determined through recovery studies, which help us understand how well our method recovers the actual analyte from the sample. This is calculated using the percentage recovery formula, giving us a clear measure of the method's accuracy. Importantly, these results must meet predefined acceptance criteria to be considered valid. Overall, ensuring accuracy is essential for reliable pharmaceutical analysis, because it confirms that our methods truly reflect the true content of the drug substances or products we're testing. For further support, please don't hesitate to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your attention and engagement throughout this presentation. If there's one key takeaway, it's that we're committed to your success and are always available to assist you. So again, please reach out if you need anything. Thank you.